Embryo transfer in humans. Introduction. First, we'll know what is embryo before knowing about embryo transfer. Embryo refers to the early developmental stage of an animal while it is in the egg or within the uterus of the mother. In humans, the term is applied to the unborn child until the end of the 7th week following conception. From the 8th week, the unborn child is called a fetus. Mammalian embryos of many species can develop in vitro from fertilization to the blastocyst stage, that is approximately 100 cell stage, but at this point they must implant in the uterus in order for embryogenesis to proceed normally. What is embryo transfer? Embryo transfer refers to a step in the process of assisted reproduction in which embryos are placed into the uterus of the female with the intent to establish a pregnancy. This technique may be used in humans or in animals, in which situations and goals may vary. Conditions for embryo transfer. Embryo transfer is needed in cases where natural fertilization is not an option or has difficulty occurring. There are many reasons for embryo transfer. They are ovulation disorders. If ovulation is infrequent, fewer eggs are available for successful fertilization. Damage to fallopian tubes. Fallopian tubes are the passageway through which the embryos travel to reach the uterus. If the tubes become damaged or scarred, it is difficult for fertilized eggs to safely reach the womb. Then comes endometriosis. When tissue from the uterus implants and grow outside of the uterus, this I have already told you while doing in vitro fertilization. This can affect how the female reproductive system works. Premature ovarian failure. If the ovaries fail, they do not produce normal amounts of estrogen or release eggs regularly. Uterine fibroids. Fibroids are small benign tumors on the walls of the uterus. They can interfere with an egg's ability to plant itself in the uterus by preventing pregnancy. Genetic disorders. Some genetic disorders are known to prevent pregnancy from occurring. Impaired sperm production. In men, low sperm production, poor movement of the sperm, damage to the testes, or semen abnormalities. These are all the reasons natural fertilization may fail. Moving on to types of embryo transfer. Once fertilization has occurred, there are a few different options available for embryo transfer. They are fresh embryo transfer, frozen embryo transfer, blastocyst embryo transfer, and assisted hatching. First one is fresh embryo transfer. Once eggs have been fertilized, they are cultured for 1 to 2 days. The best embryos are chosen to transfer directly to the women's uterus. There are certain methods to choose the best quality embryos. That will be done by the specialists of that area. So after culture for 1 to 2 days after fertilization, the best embryos are chosen and then transferred to women's uterus. This is about fresh embryo transfer. Second one is frozen embryo transfer. Any healthy embryos that were not used in the first transfer can be frozen and stored for future use. These can be thawed and transferred to the uterus. So here, healthy embryos we find after fertilization and culture. If we are not using all the embryos there for transplantation, for transfer of embryo, then the healthy embryos we find, those will be extra embryos we find, those will be frozen in cryopreservation. I have explained cryopreservation in IVF session. So those extra embryos will be frozen and stored for future use. Then when required, these can be used by thawing. Thawing means it will be stored in minus 196 degree centigrade. No, it will be thawed to room temperature and then transferred to the uterus. Next one is blastocyst embryo transfer. If many healthy embryos develop after the fertilization, it is common to wait to see if the embryos develop into blastocyst. Here, after fertilization, if we get many healthy embryos, it is common to wait till those all the healthy embryos we chose will or whether develop into blastocyst stage. Then blastocyst embryo transfer has a higher success rate than the standard embryo transfer on day 3. Usually embryo transfer is done after 3 days of fertilization. Blastocyst embryo transfer has a higher success rate than this regular method. However, recent studies suggest that it may pose risks later in pregnancy and should not always be recommended. Fourth is assisted hatching. Assisted hatching is performed in the lab just before embryos are transferred into the woman's uterus. Using a laser, the embryologist makes a tiny opening in the shell of each embryo, after which it is transferred to the woman's uterus. These are all 4 types of embryo transfers. Moving on to the procedure of embryo transfer. An embryo transfer is the last part of the in vitro fertilization process. During IVF, fertility medications are used to stimulate the ovaries into releasing healthy eggs. Then these eggs are then removed from a woman's ovaries and fertilized in a lab. Once the fertilized eggs have multiplied, the doctor chooses the best embryo to transfer to the womb. Embryos are placed in a soft catheter and then are injected in the uterine cavity through the cervix. For a pregnancy to begin, the embryo must then attach itself to the wall of woman's womb or uterus. So these things you already know. in IVF process. You can see in the picture IVF process, then fertilized embryos can be directly transferred to the womb, or they can be preserved under cryopreservation. Then the thawing step of the preserved embryos or stored embryos, then they are transferred to uterus. The transfer of pre-implantation embryos may be accomplished either surgically, that is by making an incision through the abdominal wall and exposing the uterus, or non-surgically, that is by gaining access to the uterine lumen through the vagina itself. The non-surgical method is more common. The actual embryo transfer process is the speculum is inserted into the woman's vagina to keep the vaginal walls open. Then using ultrasound for accuracy, the doctor then passes a catheter, that is a tube like instrument, through the cervix into the womb, that is uterus. From there the embryos are passed through the tube and into the uterus. The process is usually pain free and it rarely requires any sedatives or anesthesia. Embryo transfer is most commonly performed after 72 hours, that is day 3 post retrieval, after egg retrieval process. Third day embryo transfer will be performed. Next method is blastocyst transfer. It is generally performed at 120 hours, that is day 5 post retrieval. In the picture you can see the development of embryo from day 1 to day 5. Day 5 will call this as blastocyst stage of embryo. Some importance of embryo transfer technique. Transferring a fertilized embryo to a woman's uterus is an important part of the in vitro fertilization process. The objective of embryo transfer is to facilitate conception following fertilization from the in vitro fertilization procedure. The ability to produce live, young, or even mid-term fetuses from isolated pre-implantation embryos depended historically on the development of embryo transfer techniques. Embryo transfer used extensively in research with laboratory animals involves the removal of the developing embryo from the reproductive tract of one female and transferal to another. For research purposes, in laboratory animals, these, if one animal is in the reproductive stage, then removal of the developing embryo from that particular animal is done and it is transferred to another female for research purposes. Then a technique commonly used for the production of transgenic and knockout mice, transgenic animals. Embryo transfer may also be used for the production of pathogen free colonies. Experiments involving separation of maternal and fetal genetic effects are also possible with this technique. What are the risks and precautions to be taken in embryo transfer? The risks of embryo transfer. The risks of embryo transfer themselves are very low. These risks are mostly related to increased hormonal stimulation. The risks will be not directly related to embryo transfer, but the other steps like increased hormonal stimulation, it causes an increased risk such as a blood clot blocking a blood vessel. The woman can also experience bleeding, changes in her vaginal discharge, some infections, and complications of anesthesia if it is used in the procedure. Then the risk of a miscarriage is about same as in the natural conception. The greatest risk of embryo transfer is the chance of multiple pregnancies. You have read this in IVF also. This occurs when multiple separate embryos attach to the uterus. This may increase the risk of stillbirth and children born with disabilities, and is more common in pregnancies due to in vitro fertilization than natural conception. Thank you.